My podcast guest today is Dr. Thomas Seyfried, who's professor of biology at Boston College. He received his PhD in Genetics and Biochemistry from the University of Illinois back in 1976. So he knows a thing or two about genetics and biochemistry. He was assistant professor in the Department of Neurology at Yale University School of Medicine. He has received a number of Lifetime Achievement Awards, one from the Academy of Complementary and Integrative Medicine, and also the Uncompromising Science Award. I like that, the Uncompromising Science Award from the American College of Nutrition for his work on cancer. Tom has over 200 peer-reviewed publications to his name, and he's author of a truly excellent book called Cancer as a Metabolic Disease: on the origin, management, and prevention of cancer. And that is the main question we will be asking: is cancer genetic or metabolic? Welcome, Tom, to my podcast.
Oh, thank you very much, Patrick. It's very nice to be here.
Now, the opening line in one of your papers, and you've published literally hundreds, is a theory that can best explain the facts of a phenomenon is most likely to advance knowledge than a theory that is less able to explain the facts. So, as I understand it, and you can elaborate, there are two main theories of cancer. One is that it's genetic, and the other is that it's metabolic. Can you explain these two approaches and which one of them best fits the facts of cancer and why?
Yeah, well, thank you very much. This is probably the central root of the problem that we have today, is to whether cancer is a genetic disease or is it a mitochondrial metabolic disease. And because the correct answer to this, it will determine how we manage and approach this disease, cancer. So where did the fumbling around the beginning of the 20th century, Otto Warburg pretty much defined cancer as a metabolic disease based on the fact that at that time, every cancer that he looked at was big into a fermentation metabolism, meaning that the cancer cells could get energy without oxygen, and he attributed that to some defect in the ability of the cancer cells to obtain energy through oxidative phosphorylation. So he more or less said all these tumor cells are producing large amounts of lactic acid because they cannot basically respire. So he was a quantitative biochemist. He received the Nobel Prize for his discovery of the respiratory component of the electron transport chain, cytochrome C. The respiratory, at that time, it was a big advance, still is a big advance. And he claimed that cancer cells have defects in their ability to get energy through respiration. But he attempted to quantify this by looking at oxygen consumption and production of lactic acid using formulas. And there was some controversy about this. People were not totally convinced. When people looked at normal cells, or I should say, when others looked at cancer cells, they found a significant oxygen consumption, thereby challenging what Warburg had said. And I think it was around the time of Watson and Crick with the DNA structure, where the whole field shifted from the continued biochemical analysis of cancer into a genetic analysis of cancer. And because it was sexy to say that cancer involved mutations in the structure of the DNA. They looked back and they found out there was a speculative essay published in 1914 by Theodore Boveri, a famous German scientist, who completely speculated that cancer may have something to do with chromosomes. So when Watson and Crick's 1954-55 discovery started to hit the waves, they said, "Oh yeah, cancer cells have defects in the chromosomes and DNA," And the field kind of ran off with this, anointing Bovary as kind of the founding father of the genetic theory of cancer. And Warburg was pretty much thrown under the bus. They figured that these biochemists had their shot at trying to figure out cancer, and they were able to, all they're doing is bickering with each other. So the field ran off onto an analysis of gene mutations, which then exploded in the 1970s with the awarding of Nobel Prizes to people who discovered oncogenes and then tumor suppressor genes and all these kinds of genes. And the cancer field then kind of adapted this whole idea that this was some sort of a disease of somatic mutations, as opposed to germline mutations, although there are some germline mutation risk factors.
Can you explain what that means for someone who doesn't understand somatic mutations and germline mutations?
Yeah, this is an important point. So germline mutations are inherited within the genome. So we pass germline mutations on from one generation to the next, transmission inherited genetics, as opposed to somatic mutations. These are mutations that are acquired during the course of cell division or things like this. So they're not in the germline, they are acquired. And as we began to improve our technology in DNA sequencing and DNA analysis, the field began to see many, many different types of somatic mutations in every kind of a cancer, thereby linking the idea that this dysregulated growth. They have to say, what is cancer? Cancer is basically dysregulated cell growth out of control. So what is the cause of cell growth out of control? And there were, as you said, Patrick, two theories. One is that the cell growth is due to dysfunctional energy metabolism, and the other is that the dysregulated cell growth are due to mutations, somatic mutations that accumulate through wear and tear on the system. So, more and more, every major cancer has now almost have been shown to have all kinds of different genetic mutations. And the National Cancer Institute, first thing on their website, they say is that cancer is a genetic disease. And I think it's pretty well, it's become a dogmatic view. I think everybody thinks cancer is not everybody, but all the major hospitals and research centers view cancer as a genetic disease. So then they ran off in an attempt to target some of these somatic mutations as an attempt to improve therapeutic strategies for managing the disease. Turns out, though, that every cell within a tumor has a different constellation of mutations. No two cells within any kind of a tumor have exactly the same. Some tumor cells have no mutations, which they failed to discuss. And then you have now with sequencing, I think they found up to 1 million different kinds of mutations. So then they said, well, maybe not all of these mutations are really the bad guys. Let's focus on these driver mutations. So Vogelstein and others said, yes, driver mutations are the real bad guys. So we're going to start focusing on driver mutations. And now it's turning out, embarrassingly, that we're finding many, many different driver mutations in tissues and cells of our body that never become cancer. And we're also finding that some cancers have no mutations. And, this now starts to create tremendous concern about what's going on here if we have all these kinds of things.
So, I mean, to give you an example, is this a bit like saying, you take 100 women with breast cancer, and if the somatic gene theory was correct, you'd find, some commonalities in their mutations. And
that's true.
These are the genes. But actually, even the sort of hero drivers like BRCA, perhaps, would be one. I mean, one of the questions has always, interested me is, if half of women who have the BRCA genes seem to develop breast cancer and the other half don't, what's different? Why one and not another?
Yeah, well, that's a good point, because the BRCA one is a germline mutation.
Okay. So that,
and the BRCA1 is what we call a risk factor.
Mhm.
It's not because we have to deal with primary and secondary causes. A primary cause of a particular phenomenon or disease and cancer, that should always be present. In other words, if BRCA1 were to be a primary cause of breast cancer, then every woman that would have the BRCA1 mutation should develop breast cancer. But as you said, only about 50% of women that have the mutation develop it. However, every woman that does develop breast cancer from BRCA1 has mitochondrial abnormalities. So as Warburg said, all the origin of cancer comes from deficient respiration. That could happen from any number of risk factors. An inherited mutation that is somewhat penetrant could elicit the cancer, but it's a secondary risk factor. The primary risk factor is the damage to the oxidative phosphorylation. If BRCA1 does not damage the OXPHOS, that woman would have the mutation but never develop breast cancer. The question then, of course, is why would that happen? How is that possible? The highest risk factor, inherited risk factor, is the Lee-Fraumeni syndrome, which is a mutation in the so-called P53 mutation. And that's about 83%, 85% penetrant, which is very high, but it's not 100%. So, and P53 encodes a protein in the mitochondria, making it dysfunctional. So all cancer goes through the mitochondria. All the origin of cancer disrupts mitochondrial oxidative phosphorylation, whether that comes from an inherited risk factor, whether that comes from a chronic exposure to carcinogens, whether it comes from systemic inflammation, intermittent hypoxia, oncogenic viruses like papilloma, hepatitis viruses, or just general old aging. Any of these factors will damage oxidative phosphorylation, eliciting the path to dysregulated cell growth. So the gene theory is now, I've completely eviscerated this whole idea that cancer is a genetic disease, mainly because of not only what I told you, that some cancers have no mutations, normal cells have these driver mutations that never become cancer, but the nuclear transfer experiments pretty much sealed the deal on the
were they when you took a healthy nucleus and you put it in a cancer cell?
Yeah.
Just explain that in layman's terms, because I found that very convincing.
Well, you have to put those series of experiments together with the other things that I've just mentioned. Before I talk, like our ancestral people, humans that were evaluated, cancer was extremely rare. So like in Aboriginal populations, Albert Schweitzer, the humanitarian physician, in primitive African tribes, the cancer didn't exist. Eskimos, when they were first evaluated, didn't have cancer. Our closest relatives, the chimpanzee, they don't have, never been documented breast cancer in a female chimp. So you put all that together and you say, wow, there's some serious things here. But then you look at the nuclear transfer experiments, which are the most devastating observations and data against the gene theory. And that was done by a broad range of leading developmental biologists. And the nice thing about the nuclear transfer experiments is they were done in an unbiased way. These folks were not testing whether cancer arose from the mitochondria or the nucleus. They were just asking whether the nucleus of a tumor cell could direct normal development. That was their fundamental. None of them even thought about that their results would undermine the somatic mutation theory of cancer. So basically, these kinds of studies, they would have tumor cells and they would have normal cells, and the tumor cells were dysregulated in growth, and the normal cells were regulated in growth. So the idea then became, if you transfer the nucleus of the tumor cell, remove the nucleus of a normal cell and replace it with the nucleus of the tumor cell, does that now give regulated or dysregulated growth? Because if the dysregulated growth were due to mutations in the nucleus, then you would figure that the phenotype of the cell would be that of the abnormal nucleus. And surprisingly, the cancer cells were growth regulated completely. So in other words, the presence of the mutations in the nucleus were not the drivers of the dysregulated growth. On the other hand, if you took the nucleus of a normal cell and put it into the cytoplasm of a tumor cell, oftentimes the cells would die, but those that did not die became tumorigenic. So it had nothing to do with the nucleus. It had to do everything with the cytoplasm.
So it's a bit like the cytoplasm. It's a bit like an egg, where the nucleus is the yellow, and that's where the genetic instruction is, and the white is the cytoplasm. That's where the mitochondria are, which are like the energy factories of the cells. So you're basically saying if you've got a cell that's now got dysregulated growth, it's a cancer cell. It's because the engine's messed up for whatever.
So what I did, I simply went back through the literature and picked out all of these different papers that were presenting information like this, and they were repeated numerous times by the leading developmental biologists on the planet. So, and they were repeated over and over again. It wasn't like this was just a one-shot deal. All of these different studies were all consistent with each other. In the fact that the nucleus was not the driver of the dysregulated cell growth. It was something in the cytoplasm. And that goes right back to Otto Warburg. Otto Warburg said the origin of cancer is defective mitochondrial respiration, completely supporting Otto Warburg's central theory. The problem with these kinds of experiments is that people cannot believe them. There's always some superficial response when you tell them, how do you deal with the nuclear? Oh, it hasn't been repeated. What do you mean? It's been repeated by the best guys over and over again, and all different kinds of cancer models. So it wasn't just one kind of model. It's almost like I can't believe it. It's too devastating to believe, because obviously it overturns the entire cancer industry. What are we doing with all these precision medicines and all these immune therapies, all based on the somatic mutation theory? And if the somatic mutation theory is incorrect, that means the majority of what we're doing to treat cancer is incorrect. This is a very difficult pill to swallow. So if you ever hear people, you say, how do you deal with the nuclear transfer experiments if you're making all these drugs for cancer? Oh, I never heard of it. Oh, if it were true, we would have all heard of it. It hasn't been repeated. Well, these are superficial denials. If you sit down and look at the data, you come away with the understanding, clear understanding, this cannot be a genetic disease. And I think that is too devastating. I think that it's hard for an entire industry to admit that they made a mistake, because you're realizing that you're talking about hundreds of top medical schools, you're talking about a pharmaceutical industry, you're talking about federal government support, and all of that is wrong. And the answer is yes. Unfortunately, it's wrong. And how do you know that? Because in the United States, we have over 1,600 people a day dying from cancer. It's a worldwide epidemic of people dying, and there's been no major change other than anti-smoking campaigns to reduce cancer. None of these drugs that are being developed are really effective in reducing the death rate. It's perfectly predictable because it's not a genetic disease, and most of the therapies are based on the fact that it is. So it's clearly obvious. But as I said, I think it's too difficult. I think it's just a denial. I don't know.
Let's jump to the other end of the story, so to speak, and maybe work backwards, because you are developing quite a reputation in the field of cancer for helping keep people with aggressive cancers. I'm thinking of blastomas in the brain alive by the physicians who are following your science-based protocols. And obviously these have come out of a different way of thinking. That is the way of thinking that it's a metabolic disease. So, what are you doing? Why are you doing? And is it working?
Yeah. So, I mean, this now comes back to the success of managing cancer based on the fact that it is a mitochondrial metabolic disease. So this now becomes a very approachable disease, which can be managed without toxicity. And that comes from the fact that if the mitochondria are defective and the cells can't generate energy through use of oxygen, then how else do these cells get energy for their dysregulated cell growth? Because as Warburg said, and I absolutely support what he said, without energy, nothing can grow. Energy is the central issue. So if we know how dysregulated cell growth is being driven by an energy metabolism, how are these cells growing so aggressively if they don't use oxygen? And they use an ancient process of fermentation, which is energy without oxygen. So what does that mean? So what are the fuels that could generate dysregulated growth without oxygen? And that is glucose fermentation or lactic acid. They produce lactic acid. And our discovery was something that Otto Warburg did not know about, is that there is an amino acid, glutamine, which can also be fermented. In other words, energy can be generated from glutamine without oxygen. So these are the two fuels that are driving the beast. Dysregulated cell growth is from a fermentation metabolism, and the fermentable fuels are are glucose and glutamine. And we interrogated the tumor cells with every kind of a fuel to see whether they could find if there's anything other than these two fuels that could drive them. And the answer is no. So we pretty much now know how to manage cancer. We have to simultaneously restrict the two fermentable fuels. Now, we can do that. Glucose is a non-essential metabolite. We don't need glucose. We can shut down glucose from our diets. The body can make small amounts of glucose that's necessary for certain types of functions. The glutamine issue requires some drugs because there's no diet that can target glutamine. We know that water-only fasting up to 14 to 20 days can restrict glutamine, reduce it down. But it might be easier to use a drug, and we know these repurposed
essence there is that glutamine is a protein.
No, glutamine is amino acid.
An amino acid, and if we restrict, all amino acids or proteins, we may be at a lower protein, but we can't completely.
Yeah, you can't get rid of glutamine. Is an extremely valuable and important amino acid in our body. It runs our gut, it runs our immune system, it runs the urea cycle. So you can't just get rid of glutamine. You'll kill somebody. So you have to know how to strategically manipulate and target glutamine availability. And that's why we developed the press pulse therapeutic strategy. You can press glutamine, you can continue a correction. You can press glucose down because it's a non-essential fuel, but glutamine.
And that's by going ketogenic. That's by.
Yeah, well, you go ketogenic, or you can go water-only fasting, or you can do a variety of things, zero carb diets and this kind of stuff. You can push glucose way down, way down. But glutamine, on the other hand, you have to be careful with that. So you can't just force glutamine down. You're going to hurt people. So this is what I'm saying. But we know the drugs that work really, really well. And you first put the patient into a low glucose diet, a low glucose situation, which could be mostly diet, and raising the ketones, which the tumor cells can't use. Ketones can only generate energy through using with oxygen. Tumor cells don't use oxygen, so they can't use the ketones. So, and then we hit the glutamine. We pulse it. We pulse the targeting of the glutamine, and we use certain drugs to do that. 6-deoxynorleucine, which is a glutamine analog that's been used in the past, wasn't very effective because they never targeted glucose at the same time. Mm-hmm. This whole metabolic therapy works only if you target the two fermentable fuels at the same time, simultaneously, while transitioning the whole body over to fatty acids and ketones, which cannot be used directly by the tumor cells. It's not a difficult thing. I don't know. To me, it's not difficult, but apparently to the world, it seems like it's an impossibly difficult situation. I can't understand that.
So can I clarify here? So the drug basically mimics glutamine, but the cancer cell can't use it? Is that the basis for it?
Well, the drug so the cancer cell is taking in large amounts of glutamine. Mm-hmm. The nitrogen on glutamine is needed for DNA and RNA synthesis, protein synthesis. Also, the glutamine drives energy metabolism through a specific pathway in the Krebs cycle. So this glutamine gives us gives cancer cell a lot of opportunity for growth, but it works parallel off the glucose pathway, the glycolysis pathway. So you need those two fuels together, the glucose and the glutamine, which work synergistically with each other to drive the dysregulated growth. So yeah, so the cell opens the floodgates for sugar glucose and opens the floodgates for the amino acid glutamine. The drug blocks the first major enzymatic step. So the glutamine is taken into the cell and it's metabolized to glutamate, and the glutamate then is used for energy synthesis. So the drug that we use, 6-deoxynorlucine, blocks the enzymes needed to metabolize glutamine to glutamate. So you're shutting down that whole glutaminolysis pathway. So the cancer cells then become crippled. They can't grow. They can't make DNA and RNA. And then you take away the glucose and they can't make lipids or proteins. So you're shutting down, you're not only taking away the energy, you're shutting down the two pathways for the metabolites for rapid growth. So in a sense, you're completely crushing these tumor cells. And the ketone bodies help the normal cells. So all the normal cells in the body switch over to ketones, getting healthier. So we're making the normal cells of our body healthier, and we're marginalizing and destroying the tumor cells. There's nothing more elegant, nothing more beautiful than this therapeutic strategy. It works, and it works effectively if people know how to do it.
And can any doctor prescribe this drug?
Well, right now the drug, the Food and Drug Administration, it used to be approved. I don't know what happened, but it's available, but it's produced, but it's not available. I can't explain that. And I know some pharmaceutical companies are in the business of building a variant of that drug, because if they can patent the variant, then they'll make large amounts of money. But the issue, of course, is that the drug alone is not going to be sufficient to manage the majority of cancers. The drug only becomes super powerful when it's used together with glycolysis targeting, glucose targeting. So as a package deal, people have to understand that you got to shut down the two fuels together and transition the whole body over to ketones. That's the strategy.
And when you say pulsing, I mean, first of all, is it helpful at all to have a lower protein diet? And if you pulse, are there sort of periods of weeks when you're, for example, on the drug and then off? Or
no, you have to do it. In fact, if you don't do it right, you can paralyze your immune system. See, if you kill large amounts of tumor cells, you need a rapid immune system to come in and pick up the corpses. Otherwise, you're going to get an infection. You're going to end up with all kinds of other problems. So you have to be very strategic on how you target the glutamine. So, and this is what I'm saying, pulsing is just like one day with a certain dosage. And we're working out dosage timing and scheduling as we speak. So this is going to be the future of how all cancers are managed. It's just that we're on the cutting edge of doing this. And, when patients get these drugs, and it's not, there's other glutamine drugs, but we can't find anything as powerful as 6-deoxynorlucine, which is DON, D-O-N. It's an old drug, it was derived from, it was an antibiotic kind of a thing. But again, people didn't know how to use it. They gave patients too much of it, didn't work real well. You got to know how to use the tool. If you have a tool, you need to know how to use the tool. You have to shut down these two fuels and transition the body over to ketones in order to get the maximum therapeutic benefit. And when we do that, not protein, there's Pablo Kelly, who's managing his glioblastomas on a carnivore diet. He's just keeping his blood sugar low and his total calories low, and his tumor's not cured. He gets it debulked he's from Devon, England. He gets it debulked every 3 or 4 years. So we don't say Pablo Kelly is cured, Pablo Kelly is managed, and we can manage without. So he's lived 8.5 years with a tumor that kills most people within a year. And he never took any radiation or chemo or any of that stuff. And he's just doing it with a metabolic approach. And I think at some point he may want to consider some other drugs that we're working on, like some of these parasite drugs that can also target the same pathways. I mean, there's so many new things that we can do to kill cancer cells without toxicity.
And there's no need for lower protein, because you said he's doing it in a carnivore way.
Yeah. Oh, yeah. A lot of carnivore people are doing really well. As long as they can keep their blood sugar down and their ketones up, they seem to be doing relatively well.
Now,
I think.
Yeah, somebody said to me that some prostate cancers can feed on ketones. So is this a myth?
No, that's a myth. We looked at that. You can't, as I said, when I looked at the energy, when I looked at the structure of the mitochondria in prostate cancer, they were all busted up. So you're not going to be able to get energy out of ketones in prostate cancer. And as a matter of fact, I had a colleague, Dr. George Yu, a prostate cancer guy, oncologist, and he said, I don't believe you about this prostate cancer. So he said, I got some tissue from a couple of my prostate cancer patients. And he sent it to Johns Hopkins for an electron micrograph to look at the mitochondria. And sure enough, the mitochondria blown to hell. And then he comes back and he says, yeah, there's no way in hell these prostate cancer cells can use fatty acids or ketone bodies. Mm-hmm. So we found the same thing with all the cancers. They all have defects in the number, structure, and function of mitochondria, meaning that they're going to have to ferment to get energy. Mm-hmm. So all the cancer cells all these cancers all have a similar problem. They're all dependent on fermentation metabolism. Mm. So there's no unique breast cancer, colon cancer, brain cancer, bladder cancer. They're all fermenters. So once you know that, then you know how to kill them. The problem is what I said is just people can't believe it. They just can't comprehend what I just said.
No, I mean, it's very eloquent and fantastic to hear, really hopeful. Do you augment with any supplements? I remember reading about HCA, ALA, vitamin C, and so on.
I look at supplements as things that don't hurt you. Can they help you? Maybe. But all I know is these cells, without targeting their glucose and glutamine, you're not going to achieve the mission. Now, if I'm simultaneously targeting glucose and glutamine while the body is in a state of therapeutic ketosis, is it possible that we can throw some supplements in that could kind of push this along a little bit? Yeah, possibly. But let's go to the heart of the matter. Let's knock the crap out of their energy metabolism, then worry about these superficial things later on. You're not going to manage cancer with metformin or any of these smaller things that people want to use. Frank, I don't know. You can name them all. They're not going to hurt the patient. Let's put it that way. And they may have some therapeutic benefit, but they're not the big backbreaker on this whole thing. You got to simultaneously target those two fuels and transition that body over to ketones, so those fuels work a hell of a lot better. Because what we find is these glucose glutamine targeting drugs work so much better when the body is in ketosis. Mm-hmm. So and they go through the blood-brain barrier. I mean, we did all this stuff. It's unbelievable. It's actually astonishing how well this stuff works when you know how to use it and do it.
Now, hydroxycitric acid, which is the kind of active ingredient in the tamarind, Garcinia cambogia, that's sort of quite interesting in what it does to energy metabolism in the.
Yeah, I haven't looked at it, so I can't speak to things that I have not studied.
Yeah.
So when I study them, we have the best preclinical systems for ferreting out and looking at therapeutic things. And, unfortunately, these are not easy experiments because you have to try to ferret out which part of the therapeutic strategy is really responsible for the majority of the effect. Mm-hmm. So you have to run each one of these products by itself and then in combination with others. Other drugs and procedures to see whether or not who's calling the shots here, which one has the biggest effect. Mm-hmm. And then these animal experiments are not cheap. They're very labor intensive. So to really go down, as I said to my students and research staff, let's manage the disease first, big time, and then worry about all these other things later on, because everything can be perfected better. But let's give the patients a chance to live longer with a higher quality of life first, and then go back and see if we can streamline some of these things. Because if we divert our attention to procedures and drugs and things that are not the bigger, the more powerful ones, then we all these poor folks are still dying and suffering. So I want to stop the death and suffering first, and then go back and streamline the system.
Now, that makes total sense. But I do want to ask you about vitamin C, and I'm thinking here of some of the work out of Walter Longo's area. This was CRAS-positive cancer cells, where they used a ketogenic fasting mimicking diet, which seemed very effective at reducing tumor volume, or vitamin C, which had similar effect, and the two together were by far the best. And I'll just throw in that question as well. You know the work of Jane McClelland, her book, How to Starve Cancer. So at least the title sounds exactly where you're at, except there are three sides to her triangle. That is glucose, glutamine, and also fats. We put the fats to one side just for a minute. But her latest version of the book says how to starve cancer and kill it with ferroptosis. The idea being that cancer cells can't deal with the pro-oxidant effect of high-dose intravenous vitamin C. So could you say something about vitamin C in the context of this?
Yeah, well, IV vitamins, it has to be IV, not oral. The studies clearly showed that it's IV. What it does is it creates reactive oxygen. It increases ROS, reactive oxygen species, which kill tumor cells. I mean, this is the same thing that a lot of chemo drugs do, and the same thing that radiation does. It kills these tumor cells by elevating reactive oxygen species. And again, it works better if you put them on a keto reduced glucose, because then the glucose is used for the pentose pathway. You can then increase glutathione. So if you take away glucose, and glutamine, the tumor cell will become extremely vulnerable to IV vitamin C. Because you're increasing reactive oxygen species, and that's the mechanism that IV vitamin C works through. And it will work really well if you take away the two fuels that are responsible for the antioxidant capacity of the tumor cell, which is basically glutathione. So you're really, because glutamate is part of the glutathione and NADPH coming through the pentose pathway. So you're going to shut down, you're going to make those cancer cells extremely vulnerable to death by IV vitamin C if you're also targeting glucose and glutamine at the same time. So yeah, I mean, this certainly it will be therapeutic, and I know a lot of people doing it. But the problem is, if you use IV vitamin C without also targeting glucose and glutamine, you're not going to get the full power of what that therapy is going to do for you. So again, you have to know how to work all these different procedures off of each other. Now, as far as the fat's concerned, cancer cells can't burn fatty acids. Okay, so how do fatty acids provoke tumor cells? Okay, so listen, fatty acids have this capability of causing you can cause uncoupling, right? There's what we call uncoupling of the mitochondria. So the mitochondria are already defective in cancer. You throw fatty acids there, and what they do is when you uncouple the mitochondria, you bring in much greater amounts of glucose and glutamine. So fatty acids are actually indirectly supporting the glucose glutamine utilization. So some people say, "Oh, fatty acids make the tumor grow faster." Yeah, because it forces the tumor cells to use more glucose and glutamine. It's not the fatty acids; it's the glucose and glutamine. You take away glucose. Listen, we have never found a tumor cell that can survive with fatty acids alone, with ketone bodies alone. They can't live on that stuff. So they have to have glucose and glutamine. Will fatty acids make tumors grow faster? Yeah, there's glucose and glutamine available. Fatty acids can make them grow faster. But if I take away glucose and glutamine, fatty acids can't do anything. So they can't be respired. They're a non-fermentable fuel. So ketone bodies, fatty acids, all of these things that would be respired, they can't be respired. So cancer cells are dependent on fermentation. Fatty acids and ketone bodies cannot be fermented. So there you go.
Yeah, it's wonderful. I mean, we have 100 years now of the treatment of epilepsy with ketogenic diets. And I hope now people will wake up to the fact that a ketogenic diet is absolute cornerstone to drive that glucose down. And the glutamine, it seems like,
Well, yeah, we like to say the ketogenic diet, but I would say any diet that can lower blood sugar and elevate ketones would be therapeutic.
Okay.
That's why water-only fasting is so powerful.
Yeah.
I know people, you hate to say it because then people say, well, this guy's a nut. He's going to tell me I can't eat food for 14 to 21 days. But I say those people that do that seem to have tremendous success in managing their cancer.
Now, in Jay McClelland's approach, he also talks about press and pulse. I mean, the basic concept, at least to understand it, is that you weaken the cancer cell, you starve the cancer cell fuel, and then you whack in intravenous vitamin C. I'd love to ask you about dose recommendations there. And when you say press and pulse in your strategy, could you explain that a bit more?
Yeah, well, so the press pulse concept came from the paleobiologists, Aarons and West. I adapted that concept. See, the history of our species on our planet. We went through all these great extinction in the past, these extinctions, and they argued that the greatest extinctions of organisms on the planet were linked to a press pulse phenomenon. So an environmental stress on the planet would eliminate a lot of weaker species, but there was always some species that could hang on even under this stringent environmental stress. Then you couple that with a meteor strike or a giant volcano eruptions and things like that, and that would then exterminate even the hardy ones that could hang on. And it was only through a press pulse concept that you could actually cause mass population extinctions. So basically what I did was I took that same concept and said, well, if we hit them with glutamine alone, we're going to kill all the glutamine guys. And if we hit them with glucose alone, we're going to kill the glucose guys. But the glucose-dependent cancer cells, it's only when we hit them together. So you can, as I said, you can press glucose, causing a stress on the availability of fuels. But it's when you hit the glutamine, you take away their energy. So it's a combination of you want the complete elimination of the tumor cells in your body. And, what we're doing in the clinics now is we're just pouring massive doses of poisonous chemicals into the bodies of these poor folks. We're using immunotherapies, you're using chemotherapies, radiation therapies, and you're just destroying everything. But if we use a more logical strategy, press pulse, we can slowly degrade. And the idea is, when you stress the environment, the weak tumor cells will die, but then some of them will hang on. But as we pulse the glutamine, we do it strategically. So we don't get rid of all the tumor cells at the same time. We degrade them slowly. So we have kind of like a chokehold on the glucose, and then we just pulse the glutamine, and gradually we slowly degrade the tumor. So the patient comes in, the patient has cancer. Oftentimes the patient has diabetes, hypertension, has all kinds of other things. So we need to clear up the mess in the patient's body. We do that with a calorie-restricted ketogenic diet or any diet that would lower glucose and elevate ketones. And then we gradually degrade hit them with glutamine targeting drugs. So we bring the patient from a state of disease to a managed state. And then with continued dosage timing and scheduling approaches, we bring the patient back out now of the disease state. Now, think about this for a minute. If the patient has cancer and oftentimes type 2 diabetes, hypertension, a variety of other ailments, we gradually change the whole physiology of the patient. So not only do they get rid of the cancer, they get rid of the diabetes, the hypertension, and all the other what we call chronic abnormalities at the same time. So this patient now emerges as a far healthier, than they were when they first started. This is completely different than what we're doing today with these poor folks. They were irradiating and poisoning them when they come out, and about 45 to 50% of the folks that are being treated for cancer are dying from the treatments before the cancer kills the patients. So this is nuts. We have to stop doing this, otherwise we're going to continue this fiasco. This is one of the greatest problems. This is one of the greatest tragedies in medical history, is what we're doing today. It's medieval what we're doing to these poor folks.
Is the intravenous vitamin C then part of Pulse as well? I mean, how would you use that in your,
I think that could certainly be part of the Pulse strategy. No question about it. I mean, this is another strategy that works for sure.
Because everyone has a different view, whether you do it, like 3 times a week, every 3 weeks, or whether you're using 100 grams, or some go even higher than that.
Right. No, that could be certainly integrated into the Pulse strategy. Mm-hmm. As long as you're holding that glucose tight, as long as you keep that glucose under control, you're going to increase the reactive oxygen species and kill the tumor cells. The nice thing about IV vitamin C is that it can degrade. It can slowly degrade tumor cells. You don't kill them all at one time, because then this causes a reactive problem. You can die from an anaphylactic kind of a problem. Mm-hmm. So IV vitamin C is a nice add-on to this whole thing.
I can't tell you how many times I've suggested that to oncologists of clients that I've been working with, and they say, "Oh, no, you can't. The IV vitamin C will compete or interfere in some way with the chemo drug." And I've looked it up in PubMed, and in nearly every case, it just augments, it enhances.
Yeah, right. It does. It does. And as a matter of fact, the clinic in Istanbul, they use the lowest doses of chemo drugs together with metabolic therapy, and they're getting success. So, but they told me they would love to not do it at all with the chemo drugs, but it's part of the standard of care everywhere. Wow. You have to use these toxic drugs, mindlessly using toxic drugs. This speaks to a lack of knowledge on what the biology of the affliction actually is. So anyway, these are the problems we have to deal with.
So what are your results? Tell us a little bit about your results, especially with some of the very aggressive tumors you've been dealing with.
Yeah, well, I mean, we have Pablo Kelly, who's kind of now become the spokesperson for glioblastoma. He also had an IDH1 mutation. And here, the crazy thing about it is the IDH1 mutation is a therapeutic mutation. I published this. This was that nobody wanted to hear that. So listen to this. This mutation acts as a drug. Can you believe it? The mutation disrupts the glycolysis and glutaminolysis pathways, the same way our press pulse therapy works. So people who have a glioblastoma that also have this IDH1, isocitrate dehydrogenase 1, their tumors grow much less aggressively. So Pablo has one of these kinds of things. Now, here's the absurdity. So many people in the cancer field think all mutations are bad, and we have to target the bad mutations, right? So they made pharmaceutical companies that would target the product of the IDH1 mutation, which is 2-hydroxyglutarate. So 2-hydroxyglutarate interferes with the glycolysis and glutaminolysis pathways that are driving the tumor. So these companies think that's bad. So they go out and make drugs to target the very molecule that's given the patient an opportunity to live longer. Can you believe this? So then they tried some of these drugs on these GBM patients. It killed the patients faster. And yet they continue to use it's just the absurdity. To the point, sometimes I say to myself, what am I, living on a different planet? How are these people how can they lack so much knowledge on the biology of the disease they're treating, where they're making drugs that actually kill the patients faster than the disease itself? I look at this and I just keep shaking my head, yet there seems to be some momentum to build these crazy drugs that are killing these poor patients faster. I just don't understand it. My success is, we think that when we treat cancer patients with metabolic therapy. Now, we have several. My Greek colleague, Athanasios Evangelio, published a beautiful paper entitled Restricted Ketogenic Diet Therapy for Primary Lung Cancer with Metastasis to the Brain. Now, this guy from Greece, Dr. Athanasios Evangelio, understands metabolic therapy. This guy's out 9 years now. When you have metastasis to your brain from any type of a cancer, it's usually a death sentence. But we have this guy, he's out alive nine years now. The group in Istanbul, we had a woman with stage 4 metastatic breast cancer that metastasized to her brain. She was from the United States. She went to Istanbul. She was in the emergency room, intensive care for at least 3 weeks because she almost died during the trip there. Now she had breast cancer metastasis to the brain. And last year we got her and her husband were vacationing in Hawaii. That when she was in the United States, they said, "Oh, you only have 1 month to live." Well, they were obviously very wrong about that. Our newest, we have several lung cancer patients, breast cancer patients. I'm trying to write these up as case reports, right? And they always say, "Oh, it's only an N of 1. Oh, only an N of 1. Oh, only an N of 1." Are you kidding me? I says, you don't have anything in the that's working like this. Well, how come there's no clinical trials if this stuff is so good? Well, who's going to do the clinical trial? People who don't know the first thing about metabolic therapy? Who's going to pay for the clinical trial? You want a clinical trial to show that non-toxic metabolic therapy is better than all these expensive immunotherapies? Who wants to support that? So everything is quite logical. But our new paper on resolution of canine mast cell tumor. Now, this will come out tomorrow. It's going to be released for publication, a case report where we use nothing but metabolic therapy alone to completely resolve an aggressive mast cell tumor on the face of a dog. No drugs, nothing, just a massive switch in diet and completely resolve this dog. The dog died from old age, from heart failure, never. So this is the first complete resolution that we've had. I haven't been able to achieve this in our aggressive mouse models. And we're not sure yet how many of our humans can die of old age without having a recurrence of cancer. We won't know that for probably several decades, despite they're all living well with a high quality of life, but we don't know if something would recur. But in this case with the dog, there was it was complete resolution, no recurrence. Simply from metabolic therapy.
Are you saying with your patients following this approach, presumably not at the very last, moment of metastases, they're all still alive if they follow this approach, that there's no cancer that you've treated where this has been unsuccessful?
We can't. No, there's unsuccess for sure. And we don't have success is the patients were unable to get themselves into the necessary glucose ketone index that would be necessary for whatever reason. And many of our failures come from people who email me and try to adapt this when they're in hospice. They're at the end of their rope. I feel really bad, this poor guy or person, has been radiated, poisoned. I mean, he's been surgically mutilated. I mean, the poor guy is nothing but a walking bunch of bones. And then they try to do metabolic therapy and it doesn't work. But he should have never been in that position in the first place.
Now,
most of the people do well if they started at first.
Let me ask you about GKI. You may not know that I've been taking groups of, say, 18 people down to my lovely retreat center in Wales, and we've been doing a kind of 5-day fasting mimicking diet, and we want to get people to a GKI of at least 1, and they usually end up at 2. That's the glucose ketone index. So, for example, if your glucose was 4 and your ketone was 2, that's a 2-in-1 situation. If you've got 4 and 4 ketones, 4 glucose, that's 1. So we want to get from 2 to 1. Is that what you're talking about? What's the
that's right. For the what we defined it as 1.0 or below. But then when we did Pablo Kelly, he was doing really well with 2.0 and below. Okay. So we kind of, if you're down by 2 or 1, you're really you're putting a lot of pressure on those tumor cells. they're under a press. So the GKI will tell you where the press is. So and a lot of people say, well, I'm on doing keto. I said, what's your GKI? Well, I never measured it. Well, you got to measure it. Then you really know.
Yeah.
So healthy people who don't have cancer can often get well below 1. They can get down to 0.6, 0.8, somewhere around these ranges, where ketones are actually ahead of the more than the glucose in millimolar in the blood. And, we're not saying you have to stay in this zone forever. My colleague, Dominic D'Agostino, is a leader in the nutritional ketosis field. Dominic is always in a level of therapeutic ketosis. He's always down in the 2 to 3 to 1 range. We evolved as a species, in our Paleolithic ancestors, was no carbohydrates in the diet, rare, very rare. They were eating meat and berries, whatever they could find, they would eat it. These folks were always in ketosis. So we can't go back, though, and measure the GKI of our Paleolithic ancestors, but we can certainly try a Paleolithic diet and see what we get. And when you do a Paleolithic diet, you're in ketosis. So clearly.
Yeah, one of my brilliant scientists, Professor Michael Crawford, he was the guy who put DHA, omega-3, on the map. And where I live, they found a 35,000-year-old Homo sapiens in the Gower Peninsula, the Paviland man. And when they analyzed the bones, about a quarter of their diet was marine food. So, again, very, very little carbs. And by the way, everyone in the whole cancer field should be shocked and should be tremendously encouraged by what you're saying. But I've been working lately in the dementia field, and I'll give you, there's 2 parallels here. Number one, there are 300 studies now on the amyloid hypothesis, that it's the cause of dementia, which have failed. There have been 30 studies of drugs, anti-amyloid antibody drugs, that have lowered amyloid and have absolutely no clinical effect at all.
I know.
Despite that, the trend keeps rolling. And one of the things I did, everyone bangs on about ApoE4, and people get very worried if they've got ApoE4. And of course, all the good studies have changed diet or given omega-3, which is very good, or B vitamins. And recently we've discovered that the homocysteine lowering B vitamins don't work if you don't have sufficient omega-3. And the omega-3 doesn't work if you don't have sufficiently low homocysteine. But all of the good studies that have changed diet or given B vitamins or given omegas or whatever have always measured ApoE4. So I went back to see if the outcome was any different, whether you had ApoE4 or you didn't. And out of 14 studies I looked at, only one of them showed a difference. So, we're in this generation where everyone thinks it's the genes that's driving the disease, but actually, and of course, if we had another hour, we could talk about it, but we know it's what we're eating, including lots of sugar and lots of carbs. That's the major driver of cancer. That's kind of the bottom line in a way, isn't it?
And not only cancer, dementia, heart disease, cardiovascular, all these different things. They just completed the big Mediterranean diet study with 60,000, and they lowered cancer, dementia, and cardiovascular disease by about 35%. So it's not so much the Mediterranean diet; it's the absence of processed carbohydrates that are killing us. The West, we're all dying. We're all dying from the same problem. High carbo, poorly processed carbohydrates is killing us. So it's not that complicated. But it is when you're looking at a nice jelly-filled donut or a big pastry. It's like our brain is addicted to glucose, and the glucose addiction is keeping us unhealthy.
And yet here we have the wonderful cancer charities who are throwing parties where you make cupcakes and cakes and sugar-filled things, and you feed them to your neighbors to get them to give you some money to put in research. Yes, as somebody said on one of my blogs on vaccines, they said, shame there isn't a vaccine against stupidity. Anyway, just to end, your work is fantastic. You are a legend. What you say just makes sense. As a trouble, common sense is not quite so common these days. What's the name of your book? How do people find out more about your work? Is there a website to go to? How do we dig a little bit deeper into what really is a very simple and amazing?
Well, I publish most of my papers open access where anyone can read them online. So you don't have to have a subscription to a particular scientific journal. Some of them I do have in the science journals, but most of the ones you can just email my name and look at PubMed or something like that, or recent publications, and you can see the data. So in other words, the important issue here is let people read the actual case reports for themselves, see what those folks did to return their bodies back to health. Yeah, my book I wrote in 2012, Cancer as a Metabolic Disease, which is quite selling briskly, I have to be honest with you. It's very expensive. I didn't set the price on this book. John Wiley Press did. So people get angry about being so expensive. Listen, I had nothing to do with that. And John Wiley is making most of the money on that book, I can tell you that. But that takes a deep dive into the science behind what we're doing, because ultimately it's the scientific credibility of what I'm saying. Is it supported by experimental evidence? And the answer is yes. And it's getting better and stronger all the time with more and more approaches and people looking into this. But yeah, we're forging ahead. There's no question in my mind that cancer will be eventually treated as a metabolic disease with much better outcomes. It's just going to take time for the system to adapt to this new idea. And when that happens, I think we're going to finally, for the first time, reduce the death rate from cancer. It's not going to be easy because people have to really reevaluate what they're eating, their exercise levels, and all these other things. But at least it's there. It's an option that we don't currently have.
Before Linus Pauling died, I filmed him on his theory of lipoprotein A, and he looked me in the eye and he said, "Patrick, it's the logic that counts. Just follow the logic." And you've been so clear in that logic. I've learned that the thing that stops or gets in the way of the logic is the money. And unfortunately, we have a big industry that is dependent on not believing what is so obviously and apparently true. Professor Thomas Seyfried, thank you very, very much.
Well, thank you very much, too.